Great. Well, thank you to the organizers for the opportunity to present C5 Therapeutics. Carl Erickson, CEO. So C5 Therapeutics is developing a next-generation anti-C5 that we hope has transformational potential for patients with GA. We believe we can deliver significantly better visual outcomes, far fewer doses, and reduce CNV safety risk. Our story is based on significantly de-risked science, a novel bivalent anti-C5 with greater durability, biomarker technology for better efficacy, a fast, efficient phase 1-2 proof of concept design that enables potentially a launch in the 2030-31 timeframe. Very grateful to be part of an experienced leadership team and have the support of distinguished experts in the field of retina. So complements of validated pathways systemically and in retina. And the theory in GA is that hyperactive complement is mediating cell death, so anti-complement should reduce the rate of GA progression. So we're building on this theme, but really innovating in two main areas. The first is better biology, where we're really selecting only those patients where disease is mediated by hyperactive complement. And in the second, we're building better drugs that sustain full inhibition of pathologic complement for a longer dosing interval to deliver better outcomes for patients. So our innovation around biology is really to think of GA in three buckets of complement: hyperactive, where it's mediating cell death; homeostatic, where it's actually mitigating the effects of drusen and AMD and maintaining retinal health; and hypoactive, where the lack of complement in response to AMD is actually driving pathogenesis. And clearly, you don't want to treat these bottom two categories. It's really to focus on the hyper. The other key criteria we have is we're able to pre-specify the biomarker criteria for response based on learnings from rare systemic complement therapeutics. And here the idea is they must be hyper, have a major reduction in complement activity, and get back to a homeostatic level. We have multiple lines of evidence for this, suggesting at least a 50% slowing of GA and visual benefit in patients with therapeutic modulation. I'm going to share two highlights of data here now. The first, this is actually going to be presented by Dr. Dave Brown on Saturday at ASRS. But in this study, we looked at, this was a blinded, pre-specified analysis of the Regatta phase 2 study of Gemini's CFH supplementation. And we looked, without looking at anatomic or outcome data, just at the biomarkers and categorized eyes as being hyperactive and having therapeutic modulation, or hyperactive and really no impact of CFH supplementation. And with those criteria in a blinded way, we see a 49% slowing that's statistically significant at 6 months. Building on this, we've looked at real-world data and as well in clinical trials. We've been able to analyze the Chroma and Spectri lampalizumab trials along with the gyroscope focus trials based on published individual eye complement data and classify them in terms of their percentages of hyper. And what you'll notice here is that there is wide variance between trials and cohorts of patients that seemingly look the same on anatomy, and they're very different in terms of hyper composition. In a simulation, if we assume that Izervay and Syfovre each deliver 50% inhibition and slowing of GA in the hyper patients, the results from the other trials would suggest that would explain the variance seen in the pivotal trials for both approved agents, ranging from 10% slowing on average to 32% slowing on average. This gives us confidence in the complement pathway. So we're building better drugs, learning lessons from the anti-VEGFs. And really what I'll focus on are 4 key differentiation features in terms of translational criteria. And those are drug design, potency, half-life and durability, and in this case, PEG. So what we've done is we've really thought of Izervay as being the Macugen of the anti-complement class, and it's got a single anti-C5 aptamer that critically has 3 unmodified nucleotides susceptible to cleavage and degradation, loss of activity in the eye on a PEG chassis. What we've done is jump to the Vabysmo approach, where we have 2 fully optimized, stabilized anti-C5 aptamers conjugated in an antibody-like structure for greater potency and greater durability. And this is what we see in head-to-head preclinical studies. We are head-to-head more potent than Syfovre and Izervay in clinically validated diagnostic assays of complement activity in human serum. And in the rabbit PK model, we are twice as long-lasting as Izervay At 8 weeks post-injection in the vitreous, we have about the same amount of drug as Izervay reports at 4 weeks. In terms of safety and drug product, we've completed CMC at our intended GMP manufacturer, passing clinical release spec, and what we see is our dose is able to deliver full inhibition through a 12 to 16-week interval with less PEG per eye than Izervay at 1 mg monthly, which in GATHER1 had 4% CNV rates versus 3% sham. We believe the technology of biomarkers and better drugs enables 2 different TPPs, a biomarker-based superior efficacy or a broader label, both with significantly reduced dosing, 3 or 4 a year, and reduced or eliminated CNV risk compared to sham. We see both of these TPPs unlocked by the same integrated phase 1/2. In the phase 1, we demonstrate directly full C5 inhibition and complement inhibition in the eye through our dosing interval. And then in the second phase 2 portion, we are comparing and prospectively demonstrating greater efficacy in hyperbaseline patients compared to sham. For the right partner, we believe this enables at least 2 development scenarios. One is a companion diagnostic-based superior efficacy profile, and the second would be a rapid-to-market, de-risked, broader label approach with reduced dosing and safety. We're currently raising a $40 million Series A through proof of concept. Very excited about the potential here to unlock transformational outcomes in terms of efficacy for patients with GA and with far fewer doses and safety exposure. Thanks very much.